Hello, good morning again. Welcome back to the next scientific session of the Congress. That will be scientific session 6. Before starting the session, as usual, let me just remind you that we have a poster platform available. So please, you can go ahead through the posters and even engage in the discussion with the authors of the posters using the poster platform that will be open until Thursday, anytime. So the next session will be actually divided in 2 parts because of technical reasons. So the first part will include 2 talks, which will be the 2 Young Investigators Awards. And then we'll make a very brief cut, about a couple of minutes, just for technical adjustment, and then we'll follow with the flash talks till the end of the session. So having said that, let me just introduce you to our first speaker, which will be Ignacio Illán-Gala from the Hospital de la Santa Creu y Santa Pau in Barcelona. He's being awarded with the Young Clinical Investigator Award, so congrats to Ignacio. His talk is titled Cortical Microstructure and Microstructural Architecture in FTLD, Looking Beyond Atrophy. So Ignacio, please go ahead.
And to have the opportunity to present my results here today. So I'm a behavioral neurologist, and my previous research focused on using biomarkers to characterize frontotemporal dementia and Alzheimer's disease. Today, I will summarize some of my previously published works, but I will also present some unpublished data that follow up my previous research. During my PhD, I gained much experience in conventional structural MRI or magnetic resonance imaging analysis, and I explored the clinical value of a novel neuroimaging biomarker that is called cortical mean diffusivity to improve the diagnosis of frontotemporal dementia. But why do we need biomarkers? Well, biomarkers may be useful to increase the certainty of a clinical diagnosis by detecting several changes like atrophy or hypometabolism. But these changes relate to the topography of disease and not to the etiology. That's why structural MRI is considered a topographic biomarker. Biomarkers may also be useful to track disease progression, and these biomarkers are called staging biomarkers. In contrast, some biomarkers provide valuable information regarding the etiology, the cause of a given syndrome. Like, for example, Alzheimer's disease biomarkers that were presented by Alberto Yeo yesterday. By the end of this talk, I hope to persuade you that my latest research challenged the traditional dichotomy between topographic and etiologic biomarkers that you can see in this slide. Within neuroimage biomarkers, I will discuss diffusion-based imaging. These measures have a tremendous potential to unveil the earliest cerebral changes that occur in neurodegenerative dementias. First, some general principles for those in the audience who may not be familiar with magnetic resonance imaging studies. Diffusion-weighted sequences capture the movement of water molecules in our brain, and we can model this movement into a tensor that represents the 3 main directions of water movement, like you can see in the slide. If the water has a huge displacement in all directions, like it occurs, for example, in the lateral ventricle, where we have cerebrospinal fluid without any kind of restriction, then we can represent this movement as a huge or big sphere, like this. But if the water so diffusion tensor imaging is based on these principles and may provide relevant biological information of a given tissue. For example, water molecules will move in the direction of axons in the white matter, as represented in the slide. However, the movement of water molecules in gray matter will be restricted by cell membranes and other barriers. And this will be represented by smaller sphere, as we can see in this figure. So neurodegenerative diseases disrupt gray matter microstructure by damaging neuronal and glial cells membranes and increasing the interstitial fluid. At the bottom right, you see the representation of microstructural changes in the neurodegenerating cortex. These microstructural changes can be related to different phenomena, such, for example, neuroglial damage. But how do we measure cortical mean diffusivity? Well, we first determine cortical thickness with a software called FreeSurfer. In short, we preprocess the MRIs automatically to determine the PL surface, which is represented here in the slide in red. And the surface also separating gray matter and white matter, represented by this line in yellow. By calculating the distance between the red line and the yellow line, we can compute the cortical thickness as represented in the slide. Okay, sorry. Next, we pre-process diffusion weight images to obtain the mean diffusivity map, and then we co-registered the DTI, the diffusion map, to the central ribbon of the cortex, which is represented by this yellow line here. So we can study the microstructure only at the cortex, at this yellow line, and this is what we see here in the slide. Then, finally, for both metrics, we normalize our data to standard space and we smooth it using a Gaussian kernel of 15 millimeters before statistical analysis. This elegant method for the study of cortical mean diffusivity was developed by Victor Montal, a highly talented neuroscientist that is working in the neuroimaging core of our lab. All this experience with cortical mean diffusivity comes from a very fruitful research line led by Dr. Fortea, which I have the chance of having as director of my PhD together with Alberto Yeo. In previous papers, the team led by Dr. Fortea investigated cortical changes that occur in patients with AD from the preclinical phase to the dementia phase and showed that cortical mean diffusivity reflects a two-phase phenomena in Alzheimer's disease. But what about frontotemporal lobar degeneration, or FTLD? When I joined the unit 5 years ago, I started phenotyping these patients and thinking about ideas for my PhD. I remember that one day I saw one of our engineers showing results on cortical mean diffusivity in patients with Alzheimer's disease. I was impressed by the extent of cortical changes that we could observe in these patients. So I wondered that maybe cortical MD, or mean diffusivity, could be a good technique to unveil cortical changes in frontotemporal dementia. And this is how I started working on the works that I will show. So I started the most common clinical presentation of frontotemporal lobar degeneration, and this is the behavioral variant of frontotemporal dementia. This is a fascinating syndrome where patients show a progressive change in their personality with a relative preservation of cognitive function. But BV-FTD, or the behavioral variant of frontotemporal dementia, is a very heterogeneous clinical syndrome. And in accordance with this clinical heterogeneity, the pattern of cortical atrophy is also very heterogeneous. Indeed, up to a third of patients do not show the typical pattern of atrophy at baseline MRI. So this group of patients always caught my attention, and I wonder if we could be able to capture the earliest cortical changes in regions where we are not able to show or to find atrophy. To investigate the clinical value of cortical mean diffusivity in BV-FTD, we gathered a large multi-sample sample of patients with this syndrome in a paper that appeared in the cover of the journal Brain. First, we compared cortical thickness on the left and cortical mean diffusivity on the right between BV-FTD patients and healthy controls, and observed cortical thinning, which is represented in blue, in frontotemporal regions, which was an expected result. But we saw more widespread cortical mean diffusivity changes as shown in the panel on the right. These findings suggest higher sensitivity of cortical mean diffusivity to capture neurodegenerative changes. To investigate this, we computed the net effect size obtained by subtracting the effect size of cortical mean diffusivity and the effect size of cortical thickness. We found moderate to hide effect sizes favoring cortical mean diffusivity over cortical thickness in frontotemporal regions. Here we see the representation of the net effect size, net coincidence effect size. Next, we explore the group of BVFTD participants where atrophy was not evident on the visual inspection of MRI diagnosis. This group of patients is of a special interest since we usually, clinicians, struggle to differentiate between them and primary psychiatric diagnosis. Interestingly, we found that cortical mean diffusivity was also increased in the absence of significant cortical thinning. So we see green, which represents increased mean diffusivity, but we don't see a lot of blue in the maps of cortical thickness, so we don't have cortical thinning. We also found that measures of disease severity showed a better correlation with cortical mean diffusivity, as shown in this figure. Regions in green represent regions where we observe a statistically significant correlation between the modified version of the CDR sum of boxes, which is a measure of disease severity in frontotemporal dementia, and mean diffusivity. So again, correlation with mean diffusivity, but not correlation with cortical thickness on the left. In a follow-up paper, I also investigated these metrics in the ALS or amyotrophic lateral sclerosis frontotemporal dementia continuum. We know that ALS and FTD are the two ends or represent the two ends of a clinical, genetic, and neuropathological continuum. Not a long time ago, ALS patients were considered, or ALS was considered a pure motor disorder. However, now we know that up to 40% of ALS participants have cognitive and behavioral changes. The neural correlates of these non-motor changes is not clear. In this paper that we published in the journal Neurology, we prospectively recruited patients within the ALS-FTD continuum. On the left, you see patients without cognitive or behavioral impairment. In the middle, patients with cognitive or behavioral impairment. And on the right, patients with full-blown frontotemporal dementia. Patients were assessed with the Spanish version of the ECAS, which is a cognitive test. That means ECAS means Edinburgh Cognitive and Behavioral ALS Screen that was gently provided by colleagues from the Carlos III Hospital in Madrid. We observe a continuum of atrophy as shown here in the figure, but most importantly, we observe more widespread changes of cortical mean diffusivity. And again, cortical mean diffusivity showed a better correlation than cortical thickness with measures of cognitive impairment, as measured by the ECAS test. Recently, I have received my first health project grant to pursue this research line. Next, I decided to work with patients with primary progressive aphasia. These patients present with speech and language difficulties that dominate the clinical picture at the first diagnosis. Each of the three main PPA or primary progressive aphasia variants is characterized by the focal pattern of neurodegeneration that is represented in the slide. We have the logopenic variant, the semantic variant, and the non-fluent variant. And in color, you see the areas that are usually the focal areas that undergo neurodegeneration in these syndromes. We just submitted another paper exploring cortical mean diffusivity in primary progressive aphasia, and this paper is now under review in the journal Neurology. But today, I wanted to share with you some of my preliminary data on a novel imaging biomarker. To introduce this new biomarker, I would like you to think about an iceberg. Until now, we've been looking only at the little mountain of ice, and we've been ignoring what we have beneath the surface, the white matter, subcortical white matter. But we know that some neurodegenerative dementias affect not only the cortex, but also subcortical white matter. However, we don't have imaging biomarkers relating what we have above the surface to what we have below the surface. The relationship between these two components can be understood as a qualitative measure of neurodegeneration that may provide important information regarding the architecture of neurodegeneration. That's how I came up with the idea of the microstructural architecture ratio, represented here. So we can sample or look at the mean diffusivity at the central ribbon, like this is cortical mean diffusivity, but we can combine this metric with mean diffusivity at the interface between gray matter and white matter. which is represented by this blue or purple line. If we combine these two metrics for each vertex, we can compute this ratio. To explore if this novel imaging biomarker could differentiate between different neurodegenerative diseases, I gathered a sample of autopsy-proven cases during my postdoctoral fellowship at the Memory and Aging Center in the University of California, San Francisco. As you can see, autopsy-proven cases with primary progressive aphasia where Alzheimer's disease was confirmed. So here we have these maps represent the changes that we observe in 7 patients that presented with primary progressive aphasia, but where we have a definitive diagnosis on autopsy. So these are autopsy-proven cases. We see the expected changes for cortical thickness, widespread changes for cortical mean diffusivity, nothing new until here. But when you look at the ratio combining gray matter and white matter, we see widespread changes that point to a higher impairment or microstructure impairment in the gray matter than in the white matter. And so in this comparison with controls, we see in red the regions where we have more impairment in gray matter than in white matter. In contrast, when we look at in a different patient that is also autopsy proven and presented with the same clinical syndrome, but in this case, this patient have a TDP subtype type A, we see that we also have atrophy, we have mean diffusivity changes, but we don't see this predominance of gray matter impairment over white matter. Even more interesting is when we look at patients presented with primary progressive aphasia, but that have a corticobasal disease on autopsy, for example. This is a 4R tauopathy, where we find also atrophy and mean diffusivity changes. But very interestingly, when we look at this ratio, we see the inverse, the opposite pattern that we observe in Alzheimer's disease. So we have, in this case, patients have more microstructure impairment in the white matter than in the gray matter. These preliminary results show for the first time that we can detect a neurodegenerative signature that may be specific of the underlying etiology with a simple MRI. We think that these results may have important implications for the field. Imagine that in the near future, we will be able to combine plasma biomarkers with this kind of MRI studies to provide important or very relevant information regarding the etiology and also being able to stage participants. And of course, these kind of studies have a much lower cost than positron emission tomography studies, which can cost each study can cost more than almost $2,000. Finally, I would like to thank all the members of the memory unit headed by Roberto Yeo and our collaborators at the hospital clinic and the Memory and Aging Center at the University of California, San Francisco. Special thanks to Victor Montal, whom with whom I will be sharing this prize, since he teach me almost everything I know about neuroimaging, and his previous work, collaboration and support made everything I presented today possible. I would also like to thank the Global Brain Health Institute, the Carlos III Health Institute, and the Alzheimer's Association for believing in young researchers like me and funding my current health research projects. This award means a lot for a young researcher like me, and I hope it will help to move forward and my career and achieve the challenging goal of conducting first-level clinical research in our country. So thank you very much.
Also, congratulations for the award. I would like to ask you to stay online, and we will have some time for questions after the next talk. Thank you.
Thanks.
So our next speaker will be Germán Belleger from the University of Valencia. He's the Young Investigator Award of this year, and his talk will deal with Adult neural stem cells are alerted by systemic inflammation through TNF-α receptor signaling. Germán, please go ahead.
I need someone to stop sharing the previous talk. Yeah, thank you. Yeah, yeah, here you go. So, hello, thanks for the introduction. And I would like to, first of all, to thank you, the CIBERNED committee, for this award. It's an honor to me. And also to thank you for giving me the opportunity to share the project I did during my PhD in Valencia. That was finally published in Cell Stem Cell last year. So, I would like to start my talk by highlighting how lucky we are, not only as a human being, but also as a scientist, that in the vast majority of adult tissues now we know that there exists a population of what we call adult stem cells or tissue-specific stem cells that are able to generate new specialized cells of the tissue while maintaining data identity to be able to generate new cells in the future. So these cells are responsible of the cell turnover under physiological conditions and also to fulfill the cell demand for tissue repair. And these functions require a fine-tuned balance between cell renewal, proliferation, and differentiation that is greatly regulated by what we call the stem cell niches and all the signals the surrounding provides. Therefore, these cells are not only the natural source of regeneration, but also a great opportunity for us to understand how to produce new cells. And it becomes evident that the more we know about these stem cells, the more doors will open for future therapeutic opportunities. So with this philosophy, in the lab of Professor Isabel Fariñas, we are interested in understanding the function and regulation of adult neural stem cells that are localized in the supramandibular zone, which are responsible to generate new immature neuroblasts and migrate to the rostral migratory stream to the olfactory bulb, where they integrate as new neurons. But before you can understand and study how these stem cells function and are regulated, you need to be able to identify them. So when I joined the lab already 10 years ago, all that was known is that there exist GFAP astrocytic cells that presented slow cycling abilities that were able to generate fast proliferating progenitors that divide for several rounds and generate new neuroblasts. But these pictures soon changed. In 2014, other groups described that these stem cells can coexist in different states, and you can find cells with a more activated phenotype and are able to proliferate, while other stem cells remain quiescent and do not proliferate. In 2016, different groups were already able to isolate and characterize different cell groups that display similar features of activation and quiescence by applying FACS techniques and combining these with different reported mice and combination of cell surface markers. Furthermore, the single cell era started, and this brought more even greater heterogeneity at different groups identify at the molecular level cells that display different degrees of quiescence, and you can find cells that looks more dormant, while other cells are like in an intermediate state between quiescence and activation, and showing a more shallow quiescent state or a primate for activation state. So it became clear at that moment that we needed new tools to be able to identify properly our cells, to be able to study them. So for that, we sat down and decided to design a new protocol, also based in FACS technology, following 3 criteria. First, that it could be used in any genetic background, so avoiding the use of reported mice, that it allowed the identification of the cell population that was resolved by single-cell approaches, and also that it could be combined with classic purchase paradigms to conciliate with classic experiments. And with this, we designed a strategy. This is the overview of our strategy that basically is based in a first exclusion of non-neurogenic related cells to later stratify our neurogenic lineage by the expression in one side of the origin as an associated marker, and in the other side, CD24 as the progression of the neurogenic lineage. And in the middle, the use of EGFR as a marker of activation and CD9 levels to distinguish between non-neurogenic and neural stem cells. So with this strategy, we've been able to identify up to 8 different populations, the astrocytes, 2 different populations of quiescents, the active neural stem cells, 2 populations of neural progenitors, and 2 populations of neuroblasts. And due to the length of the talk, I cannot go into detail into this, and you will need to believe me, but we were very happy to see that our isolated populations show the great coherence with was expected for them in terms of the transcriptome to a similar extent of the single cell approaches, with the in vivo dynamics, both during regeneration of the supraparenchymal zone or during their cycling state in homeostatic condition, and also their ability to generate neurospheres in vitro, which is a gold standard in the neural stem cell field. Of notice, I would like to say that with this protocol, for the first time, we can isolate cells in shallow quiescence that are not cycling in vivo, resemble the ones that were described as single cells, and show an intermediate state between activation and dormancy. Furthermore, applying this, now we know that these cells are able to generate neurospheres when placed in vitro, despite they do not proliferate in vivo, and that these cells coexist in our cultures for the long term of the culture, showing a slow cycling kinetics and a transcriptome very, very similar to the one from the primate cells that we obtained from the supraparenchymal zone. Opening the opportunity to understand and study this new described state. So now this has been recently published in a step-by-step protocols that now anyone can use, and I couldn't go into detail, but I would be more than happy to discuss this later with anyone that is interested in using it or wants to apply a protocol like this in the system. And now, of course, if you can know how to identify your cells, you can go back and see and try to understand how they function, how they are regulated. So we were interested in how the regulation from quiescent and activation and the other way around is regulated. And one of the things that we realized is that quiescent fractions are enriched in gene signatures related to inflammatory response, not only in vivo, but also in vitro. It is well known that the neural stem cells are regulated by many signals that come from the surrounding area. But not only this, also from signals that come from long distances through the blood vessels, and also from neurons or the choroid plexus or the cerebrospinal fluid. Additionally, there is increasing evidence that stem cells in other systems are sensing the injuries in other parts of the body, as it has been described for the muscle and the hematopoietic system. For example, if you injure one muscle in one side, in the other side, the resident stem cells respond to this injury and are primed for activation. So we thought that this is a nice scenario to test whether quiescent neural stem cells are regulated, could be regulated by long-distance signals, and especially because it's known that, and we thought that systemic inflammation could be a good model to test because it's well described that it activates microglia and also can cause a secondary neuroinflammation in the region. So to test this possibility, we combine our protocol with mice that were injected and with LPs to induce systemic inflammation. And to test whether this could be activated in our cells, we injected EDU 1 hour before sacrificing the mice at different time points. And what we could observe is that there is a transient activation of neural stem cells that enter to cell cycles after LPS injection, but this activation is it's reduced at the later stages. Interestingly, this transient activation even turned into a high production of new neurogenic cells. In fact, it's the opposite. What you see is a less production of the latest stages of the neurogenic progression, less progenitor and less neuroblasts, while you increase the number of quiescent stem cells, a population that do not proliferate at all, not even after inflammation starts. So we wonder whether this transient activation could be coupled to a mechanism of going back to the quiescent. So to test this possibility, we apply different regimens of EDU incorporation to mark proliferating cells. And here you can see that if you inject EDU and sacrifice the mice after 1 hour, what you label is the proliferating states. You can see active neural stem cells, progenitors, and neuroblasts to incorporate the EDU. If you increase the number of doses of EDU and inject them up to 7 times during 12 hours, what you can see is that a higher proportion of cells incorporated the cells, meaning that other cells enter proliferation as well. But the quiescent fraction still do not incorporate EDU, meaning that they are very reluctant to proliferate, even they are not slow cycling proliferative cells. But interestingly, if we wait 12 hours after the last injection of EDU, what we can observe is that many cells that proliferated now are arriving to the later stages of the neurogenic lineage, which makes sense. You are producing new neuroblasts, but also you can observe that cells that proliferated now are going back to a primate state. So with this, we observe for the first time that it looks like that neural stem cells under an anesthetic condition cycle between a primate and an active state. So what about systemic inflammation? So if you induce systemic inflammation by LPS and apply the same paradigm, what you can see is that you have activated your cells, you have more cells that had proliferated in the first 12 hours, but now many of these cells are going back not only to a primate state, but also to a deep quiescent state, something that never happened in anesthesia. So this could be demonstrate that cells after activation can go to a deep quiescent and explains why we observe more quiescent stem cells when inducing systemic inflammation. And what's the mechanism of this? So if we look into detail into the inflammatory response characteristic of the quiescent stem cells, what we see is that the top upregulated term in this transition is TNF-α mediated signaling pathway. And interestingly, this cytokine is highly upregulated 24 hours after LPS injection in the superendomal zone. Who is TNF-α? TNF-α is a very well-known cytokine that has been studied in many systems, also in neural stem cells with very contradictory results, that it binds to two different receptors and has many different functions. So we tested the possibility that this signaling and so these two receptors could be mediating this mechanism, and indeed, using different in vitro models that I'm not showing today, but also genetic knockouts for both receptors, we could demonstrate that TNF-α1 is required to mediate the return to deep quiescence. If you don't have TNF-α1, you can see that cells got activated, but they do not go back to a deep quiescence state, while TNF-α2 is completely required to start the mechanism. In fact, if you don't start this mechanism, you don't see cells arriving, going back to quiescence. And with this, I would like to finish my talk with what could be the potential relevance for therapy of this mechanism. And now we demonstrate that if you treat mice that are recovering from chemotherapeutic treatment and they try to regenerate, if you treat during this regeneration process your mice with a combination of anti-inflammatory drugs, what you see is that these mice do not recover as well as their control group. Interestingly, during this regeneration, there is a peak of upregulation of TNF-α that is blocked by the anti-inflammatory drugs. Additionally, if we apply the same regeneration paradigm to TNF-α2 knockout mice, what we can observe is that these mice are completely fine to restore neuroblast production. It's not a problem to regenerate the production. But what you see is that this mice has lost stemness. If you test these cells in vitro for neurocell formation and for multipotency, what you see is that these cells expand worse and differentiate less than the wild-type controls, meaning that this mechanism helps to protect the stemness during regenerative processes. With this, I would like to conclude with the highlights of the paper. That is that now with this protocol, shallow quiescence can be prospectively analyzed in vivo in the superendomal zone. That neurocell culture retain a population of a low cyclic primate-like neural stem cells. That systemic inflammation reaches the brain and TNF-α preserves neural stem cell stemness, and that TNF-α1 and TNF-α2 coordinately promote the reversible activation of primary neural stem cells. With this, I would like to thank especially Isabel Fariñas for the opportunity she gave me and for being my scientific mom, José Manuel Moranta Redolat for being my scientific dad, and also all other my landmates, especially Ana Domingo and Pere Duarte, who helped me a lot with the project and for many other reasons. Thank you very much for your attention.
Thank you very much, Herman, for a very nice talk, and also congratulations for the award. So we have a couple of minutes for some quick questions. Again, if anybody has interest in making a question, please do so in writing using the platform, and I will be able to transfer the questions to the speakers. And also, if anybody in the room wants to make a question, we have a microphone available, so we can do these questions. I'm not getting any questions yet. Let's wait for a little bit to see if there are any questions coming in. All right, no questions. No question from the room either. So, yes, there is one question from Manuel Guzman.
Yeah, this is for Germán. Awesome work. Congratulations. Have you only tried LPS as a model for systemic inflammation damage, or do you believe that with other stimuli or the peripheral stimuli you could also have similar effects?
I would expect that because it has been described that many situations of our normal life can increase and produce at least low inflammation. So I would expect that any injury that can be sensed by the surrounding area of the neural stem cells can modulate and regulate these cells. What's the purpose? We don't know yet, but I would love to test that.
Thank you, Herman. Any other questions, either from the online audience or here? Okay, seems that there are no more questions. So as I say at the beginning of the session, we need to make a brief break now because we have some technical adjustments to do. It won't be more than 2 or 3 minutes. So please stay online for the rest of the session. And I would like to thank again Ignacio and Germán for their very nice talks. And again, congratulations to both of them for the Young Investigators Awards. So we'll be back in 2 or 3 minutes.